Good day to my lecturer. My name is PetrisyiaAnakBenji, an undergraduate student from the Bachelor of Plant Science program at the Faculty of Science and Technology, UNIMAS. Today, I will be presenting my individual mock research proposal for course STT2393 titled Effectiveness of Aloe Vera Gel as an Edible Coating to Extend the Shelf Life of Harvested Amaranthus Tricolor L. This proposed study is structured under the guidance of my supervisor, Professor Dr. Muhammad Effendi bin Wasil. Amaranthus tricolor is a highly nutritious, fast-growing leafy vegetable. However, its post-harvest management is heavily limited by its extreme perishability, often lasting only 24 to 48 hours before degrading. Because the leaf possesses a high surface area to volume ratio, they undergo rapid respiration and transpiration rates, causing immediate moisture loss, wilting, and severe yellowing. To address the challenge, without chemical additive, my research proposed a non-thermal sustainable preservation strategy, utilizing a natural, biodegradable aloe vera gel edible coating to act as a physical barrier. To evaluate this, I have set four specific objectives for my project. First, I want to assess how effectively the aloe vera gel coating minimizing physiological weight loss and transpiration. Second, to identify the optimal concentration among 25, 50, and 70% variation to protect leaf color and texture. Third, to investigate the synergetic effect of combining this coating with ambient versus cold storage environment. And fourth, to directly compare the overall shelf life and time to senin sites against the uncoated control group. Based on the literature, edible coatings form a semi-permeable membranes that restrict oxygen uptake and carbon dioxide release, creating a beneficial modified atmosphere. Aloe vera is an exceptional material for this because its inner clear gel parenchyma contains rich bioactive ingredients with natural antimicrobial and antioxidant properties. Furthermore, while standalone refrigeration at 4 degrees Celsius slows down enzymatic metabolism, it lacks a barrier to stop rapid leaf transpiration. Combining the aloe vera coating with cold storage creates a dual action approach that prevents desiccation while retarding biochemical breakdown. For my methodology, I will pursue fresh uniform amaranthus tricolor sample from a local farmer in Kota Semarang, Sarawak. I will handle the aloe vera processing manually in the lab by cutting thick outer leaf close to the base, draining the bitter yellow latex, and scooping out the clear inner gel. The raw gel will be blended, filtered through a fine muslin coat, and stabilized using ascorbic acid and vitamin E to prevent room temperature spoilage. I will then prepare three individual working concentrations, 25%, 55%, and 75% volume per volume using distilled water, maintaining a target pH of 4.0 to 4.5. My experimental framework will follow a completely randomized design. I will set up four distinct treatment groups, each executed in triplet to ensure data reliability, an uncounted control group T0 and three gel treatment groups T25, T50, and T70. I will submerge the vegetable sample in their designated solution for exactly 2 minutes, followed by 30 minutes of air drying to allow the cohesive edible film to settle. Once prepared, the individual sample will be divided into two storage trials: ambient storage at 28 degrees Celsius and cold storage in a lab refrigerator at 4 degrees Celsius. To collect accurate data, I will monitor the sample at strict 24-hour intervals until they fully deteriorate. I will track physiological weight loss or PWL using an electronic analytical balance and calculating the percentage change with the formula shown on my slide. Changes in visual quality and wilting will be cross-referenced daily against a standard 5-point visual scale. A score of 5 indicates pristine, vibrant green leaves, while a score of 3 marks the exact limit of marketability. The cutoff for shelf-life will be determined by the total number of days a sample successfully maintains a score of 3 or higher. I anticipate that the 50% and 70% concentration will perform best because Amaranthus leaf possesses a very high stomata density. The thicker physical barrier of a 70% coating will maximize moisture retention without causing unwanted anaerobic fermentation. Ultimately, I expect that combining the 75% aloe vera plume with cold storage will effectively preserve chlorophyll and prevent tissue aging, extending the vegetable frost harvest shelf by an individual margin of 3 to 5 days over the control group. To ensure the successful execution of this project, I have structured my research across a fixed 12-week timeline as shown in the Gantt chart. Week 1 and 2 will focus on sample measurement and lab setup. I will dedicate weeks 1 to 4 to extracting the aloe vera gel and formulating the coating variation. The depot treatments and baseline storage experiment will run from week 3 to week 6. Following this, a strict 4-week data collection window will take place between week 5 and 8 to closely monitor daily physiological changes. Data analysis will occupy week 7 to 10, leading directly into final report drafting and revision through week 12. This systematic workflow will keep my individual studies strictly on track. That concludes my representation. Thank you very much for your time.